So we have another Stanford speaker, Scott Boyd, who is next. And one thing I didn't know about Scott is his pedigree, a number of obscure institutions, starting when he was a Rhodes Scholar at Oxford, and then an MD from Harvard and a PhD from MIT. But after that, he somehow decided to come to Stanford and did both residency and fellowship and postdoc at Stanford, and he's now a professor of pathology. He is Stanford Professor of Food Allergy and Immunology, co-director of the Sean Parker Center for Allergy and Asthma Research, and his laboratory uses a lot of serological analyses, DNA sequencing, single cell experiments, etc., to study B cells and what they're doing in vaccination, infection, and other settings. And it's a pleasure to have him here today. Thanks, Scott.
Thank you very much, Holden. And it's great to be part of this symposium with so many collaborators and friends, and really to hear the great science continuing despite all the other things happening in society right now. So I was going to talk about some approaches we've been taking to get a better look at the B cell populations in people responding to vaccines and infections, and to be able to better probe B cell memory and really see what's happening at a scale closer to what the reality is in each of our bodies. So some of the main questions that motivate us in this, and you know, my lab has really worked on human studies from our very beginning, is to understand when we're responding to infections or vaccinations, really which antigens in the mixture that you might be exposed to are the B cells responding to, what's different between vaccination and infection, and you know, what are the key epitopes. How do antigen specificities differ between B cell populations based on their phenotype, or where the antigens encountered, what the context is. In many microbial infections or vaccination contexts, there's a real interest in what's the effect of the first memory populations that you form based on, for example, the year in which you were born, or which variants of a virus you were first exposed to. And tying into that is also, what are the factors that influence how broadly you can recognize new variants that come up in future? And so that's key for many different vaccine topics, as has been mentioned in the symposium today. In terms of the mechanisms of how B cell populations adapt to changing antigens, how much does somatic mutation or the ability to mutate antibodies affect what you can then recognize later, and how is that regulated? And then which clones end up being the ones that are secreting the most antibody, which last the longest, which really provide longer lived protection. So in this context, it's, I think, you know, taking a step back, we really have not, you know, in the whole global effort of science and immunology, haven't really characterized that many human antibodies in full molecular detail in terms of knowing their sequences, knowing their structures, knowing what they bind in terms of different antigenic variants. So even, you know, after all the mass effort of coping with SARS-CoV-2, the new virus that appeared, you know, one of the major databases summarizing all the monoclonal antibodies for which we have sequences and binding specificity, usually against just one or two SARS-CoV-2 antigens, there's only about 13,000 total monoclonal antibodies there. And similarly with influenza, you know, if you look up how many fully defined heavy light chain pairs with known specificity are there in the literature in different databases, there's only, you know, and it also numbers in the thousands. And if we try to examine antibodies where there's really a good quality structure, where you know exactly what the epitope is and how the antibody binds, there's also sort of a starvation of data with in the range of, like, you know, 3,000 to 4,000 sequences known. When we compare that to what each of us is actually doing when you get exposed to an infection or a vaccine, you know, each one of us, when we get the flu vaccine, hundreds of clones respond from your B cell memory populations, and in some cases thousands. And you know, and this is sort of lower estimates based on what we actually see showing up in the blood, rather than in the other lymphoid tissues in the body. So each one of us is probably harboring, you know, similar numbers of ways of responding to antigens that would be around the same scale as the whole world literature of the characterized ones. So we think there's kind of a starvation of data related to the specifics of how people are responding to pathogens or vaccines with their antibodies. And especially in this day and age where there's been such improvements in some of the analytical approaches, machine learning methods, AI, and so on, those are things that are generally very data hungry, right? You know, all the natural language models that were trained on all the text in the internet those are trained on, many, many, many orders of magnitude greater than what we have to work with in biomedicine. So we think this is an area where there's a lot of room for improvement and ways that we might actually be able to better understand the details of how we're each responding to vaccines or infections, and maybe be able to help us make better predictions about what kind of antibodies are going to be useful for different variants in future for a virus, for example. So members of my lab have been working quite a bit to try to scale up some of the methods that we have for identifying antigen-specific B cells and characterizing the antibodies that they make in ways that are practical and sort of affordable to actually do in an academic lab setting. And so the main thing that we've been doing is developing panels of multiple antigens, often variants of a particular virus. So for example, these are different influenza HAs from different strains of flu. And we take each one of those as a pure protein and a biotinylated protein and make a complex of it with streptavidin. And the streptavidin has a DNA barcode or DNA tag on it that will tell us what the antigen was that was in that complex. And there's also a fluorophore on it. So we can make these kind of complexes for many different antigens and then pool them into one big pool. There aren't enough fluorophores to tell all the antigens apart, but the DNA tags will tell us uniquely which antigen a particular B cell bound. So that's the next step, is we combine the complexes of antigen with the B cell containing sample, like from the blood or from human tissue of other kinds, sort out any of the B cells that bind any of the antigens in the pool. And then we do single cell sequencing, where we get for each B cell at a single cell level gene expression, BCR heavy and light chain sequences paired, so you can actually make the native monoclonal antibody. And then also we sequence the DNA tags associated with the antigens that bound to that B cell. So it's very nice if you have a B cell that binds, for example, a handful of these antigens, but not the rest. The DNA tags associated with those ones are the ones that are going to show up in this part of the single cell sequencing data. So it means that for every one, you actually get this, here's the BCR, here's the antigens it binds or does not bind. And that's the kind of data that we really haven't had for studying human immune responses, except on quite a bit smaller scale, where you had to make the monoclonal first and then test it in a panel of immunoassays. So, I'm going to show some data based on what happens if you look at human responses to a standard seasonal influenza vaccination with this sort of approach. And these data come from really nice collaboration with Scott Hensley's lab at UPenn and some other collaborators. Andrew Ward and Ian Wilson's group are doing structural characterization of a bunch of the antibodies in this case. We basically took day 7 samples from people who had received a seasonal flu vaccination in 2022 winter. Mostly these were adults, typically younger adults in this particular case. And for this, we were trying to probe really how broad is the memory B cell populations in people against a wider range of influenzas than we really usually encounter. So we included a bunch of avian influenzas, ones that were associated with previous bird flu infections, and some standard H1, H3 antigens as well in this experiment. So from the just standard PBMC samples from these individuals, we were able to sort out the B cells that bound any of the antigens in the panel, as well as a whole bunch of the plasmablasts that show up at day 7, quite enriched for the specific cells. And so in this initial data set, we had like around 11,000 plasmablasts and 1,600 memory B cells. We're able to assign specificities to most of the memory B cells. We've also been making antibodies from a bunch of the plasmablasts where they don't have enough BCR to actually sort them for antigen specificity. And in parallel, we also studied resting memory B cells from and also a large set of healthy blood donors with a wider age range incorporated in them, so multiple decades of age. So fairly standard experiment for sorting the B cells. This is the antigen panel that we used. So we basically have a couple H1s, a couple H3s, and then some, as I mentioned, bird flu kind of antigens like the H5, but also some that have only really showed up in avian influenza. So people have really never been infected by these in this experiment. These are all made with a Y98F mutation, which is helpful because it decreases sialic acid binding by the HA, so they don't stick to just every B cell indiscriminately. And I'll just orient you a bit here. So in influenza, there's 2 major phylogenetic kind of divisions. There's the group 1 viruses, which include H1, H2, H5, as well as some of these other ones that are predominantly avian ones. There's also the group 2 antigens, which are basically, or group 2 viruses, which are different from the group 1, and they include the H3, which is the other commonly circulating virus type that we have in our population. It also includes the bird flu H7, the bird flu H5 is in group 1. So with this, if we do flow cytometry of the pool of antigens, these are the B cells that bind. We usually make the fluorophores have 2 different colors so we can pick out the double positive cells. And we also sort the plasmablasts. And this is what you get if you look at the DNA tag counts for different antigen combinations in the B cells that you sorted in this manner. So if we look at the ones that are 2 different varieties of H1, we see a correlation of many B cells being specific for both, some B cells only binding one and not the other. The same thing is true for H3 antigen variants. And then if we look at an H3 versus an H1, they're mostly mutually distinct populations. A little bit of cross-reactivity, I think, is seen in the diagonals for each of these populations, but we do see some that are actually B cells that are binding both. And I should say, we also cast a wide net here. So we didn't exclude the non-class switched memory cells because we want to basically study as much of the whole response as we could. So when we look at the big overview of the data that we get from this experiment, each of these heat maps summarizes the specificity data for over 1,000 B cells. So each row is 1 B cell. And where you see red in the heat map, that tells whether or not it bound each of the antigens that you see indicated on the columns at the bottom. So the ones that bind group 1 antigens, which are the sort of left half here, we can see there's actually quite a bit of cross-reactivity amongst group 1 binding B cells with strains of virus that you've never actually been infected by. So there's sort of an underlying recognition of shared epitopes, even much more widely than just within the variants of H1, for example. The same is true if we look at the group 2 antigens, including H3. So there's a bunch of cross-reactive ones that recognize both of the H3s that we include in this panel, but also a number of other group 2 antigens that are part of this. And the influenza B binding B cells that were in the experiment, they're pretty distinct from both. When we look at memory cells of blood donors, that's this panel. And then here, the vaccinees, what we see at day 7 after vaccination, we see pretty similar patterns. But as I'll show when we quantify it, we see somewhat greater breadth of the B cells that are binding to the antigens in the post-vaccination state. So in this single kind of experiment, we get to look at basically close to the order of magnitude of the number of well-characterized binders that there are in the whole literature otherwise right now. So I think that between this kind of work and similar efforts going on in other labs, the amount of total antigen-specific BCR sequence data we're going to be able to evaluate is going to be increasing rapidly, in the next few years. We had, of course, had to validate these assays, so we also made a bunch of these as monoclonals in collaboration with the Hensley lab at UPenn, and we've tested them with, Luminex, just standard binding with the monoclonal antibodies, and the results correlate quite nicely with the DNA tags. So we have the indication this is a reliable approach. Here you'll see that these are basically historical H1s, so an extended panel, historical H3 antigens from human populations that were, human infecting viruses for many decades going through the past. Some historical B antigens. This is our panel that includes the a couple H1, H3s, but also all the avian ones. And so essentially, you can see some antibodies are able to bind very broadly. These are generally ones that recognize the stalk of the HA, and other ones are relatively specific to H3 or H1. So for example, these ones are H1 binders, really binding mostly the H1 panel members, H3 binders, flu B binders. And then we did find a bunch that are actually cross-group binders. So these are amongst the broadest of the flu-specific antibodies that people have in their immune memory. And a lot of these are stimulated by vaccination. So we have a paper characterizing some of these in detail that's currently under review right now. But it shows that there's actually quite a bit of cross-group, relatively broad influenza-specific memory present in everybody at baseline that also gets stimulated by vaccination. This is just another summary of the binding patterns. So these are the group 1 antigens, and you can see that there's a lot of correlation of B cells that bind the different group 1 members. The same is true for the group 2 members here in this upper corner, but there's also some antigens that seem to be bound relatively broadly across different B cells. So some of them have very shared epitopes, we think. When we summarize what are the proportions of cross-group binding antibodies or the patterns that we see, in general, if we look at the resting memory versus the vaccination stimulated ones, we see that there's actually some increased proportion of broad group 1 binders in the resting memory and a relatively higher proportion of group 1-2 cross-reactive B cells that are stimulated by vaccination. So it does seem like there's some shift in the populations, and that's stuff that we're continuing to study in other cohorts going forward. So this just sort of summarizes that. If we look at the cross-group binders, we see more of them in the vaccination day 7 group compared to the resting memory blood donors. Something else that starts to come out of having the larger scale of data in this sort of experiment is that we can really start to recognize much more comprehensively what are the common patterns of antibodies that people use to recognize particular types of influenza. So if we look at the gene composition, like, which V genes are being used in the heavy chain and light chain, what do the CDR3s look like? There are a bunch of patterns that show up in multiple people. So they're public antibody types, and they may not have highly similar CDR3s, but they often have, motifs or particular pairs or single residues that are actually really important that you can recognize if you have enough data to detect them. The other nice aspect about this is that if you have the binding data for a panel of antigens, you can actually start to sort out the B cells based on, forget about their sequence from the start, but just see which ones bind similar patterns and variants within the pool. That can also help to narrow in on which epitopes they might be recognizing. So it's actually quite, simplifying in terms of understanding the relationship of the sequence to the structure, and then the function in terms of which ones are actually going to bind head epitopes that might be neutralizing ones, for example. So basically, we see a bunch of patterns of H1 binders that are pretty characteristic, a different set of patterns for H3 binders. If you look at the ones that use kappa light chain versus lambda, you see different sets of genes. So these are some of the fundamental ways that people just through evolution have evolved to actually recognize different influenza antigens. And then if we look at the especially interesting ones, like the cross-reactive binding B cells, we see a bunch of really prominent patterns. Like, for example, there's a variety of these B cells that use V1-18 as their heavy chain V gene. And we really see the characteristic frequency of binding to all these different influenza HAs. Similarly, for some other ones using particular other V genes, like V333, V353, and V6-1, a few of them have been reported in the literature already. But we think that the description of those is actually very incomplete. Like, there's lots of variations on those binding patterns that are actually accounting for a greater breadth than has been appreciated up to now. So this is similar just with the lambda light chain using ones. So this is getting us closer to actually really being able to predict the binding of a new antibody if we hadn't seen it before. So we've been continuing this work with expanded panels of historical HAs to get a much better idea of like which of the previously encountered ones in human populations do you recognize, and how does that relate to what you saw when you were a child or what you were first vaccinated with, what kind of imprinting you may have had in your B cell memory formation. And we've also been putting a lot of effort into analyzing B cells that recognize the new H5 bird flus that have arisen in the last few years as the virus types have come to North America. So the lab members, especially Oliver Wirtz and Artem Mikolov, I mentioned particularly, have been working a lot on also expanding this to really give us a tool to interrogate much more broadly the memory against other viruses at the same time. So not just influenza, as I focused on for the beginning of this, but this is a panel that we've used now in sorting a bunch of human organ donor samples that my lab had collected, and also look at healthy blood donors where we can get abundant blood products. But we can basically interrogate the B cell memory for influenza, SARS-CoV-2 and other coronaviruses, other respiratory viruses, sampling of enteric viruses, herpes viruses, childhood vaccines, and other antigens, including we have a lot of interest in food allergy in my lab as well. You can basically sort out the B cells that bind all these all at once and really study the person's immune memory, what differs between viruses, and so on. So that's something we're quite excited to pursue in a lot of different contexts, like what changes with age, what happens when you're first exposed in childhood, and that sort of thing. Now, this is some data from that large panel with, I think, 50-some antigens in it that Oliver carried out. And so you can see, for example, these are all the SARS-CoV-2 and some other coronavirus binding B cells. These are a bunch of the different influenza binders and many others. So this is an approach that we think is applicable to many different projects in immunology. And by looking at these kind of data, we start to recognize other patterns that haven't been as clear from separate individual studies in the literature. For example, if we look at the rate of somatic mutation, so the additional mutations that get added to B cells as B cells receptors as they're stimulated, the highest mutation frequencies that we see are basically for things like the spike antigens or the head of the influenza HAs. If we look at the stalk portions of these antigens, we find that in general, both for SARS-CoV-2 and for influenza, the stalks stock-specific B cells seem to get less mutated, even though those are portions of the antigens that are actually relatively well conserved and are stimulating the B cells many times. So there's sort of some fundamental differences in, depending on which epitopes your B cells recognize, how much they get affinity maturation, how much they get mutated over time. So that was sort of a surprising convergence of findings from two different viruses in terms of the coronaviruses and flu. So, so this is where we're headed in terms of being able to better probe human vaccine responses, better understand what's happening when you respond to an infection. And we think these tools are actually going to really allow us to collect data that's more on the scale of what the human immune system has been doing all along, when we were able to just get much more sort of tailored data sets previously. I also wanted to talk about something that was a surprising finding that came out of some of this work. You know, I present these antigen panels as though it was just a matter of, you know, buying or making the antigens and putting them together. But lab members have done a ton of work to troubleshoot and actually get these things to work and have all the antigens ready at the same time, which is a real technical, you know, tour de force. But as part of troubleshooting some of these antigens, we've actually learned interesting things about some of them. So this, I give special credit to Taylor and Oliver and also Artem for this set of new findings here. So we were studying the HAs of the new bird flu H5N1 hemagglutinins. And what we had noticed is basically if we tried to stain B cells with the H5 HA from the new antigen, we saw that, for example, if we do staining with a regular H3 HA, we get a bunch of positive cells in a typical frequency range, like, you know, some per 10,000, for example. But when we tried to do the staining with the new H5 N1 HA, in this case from the American Norwegian isolate of the virus, there's a ton of high background, right? And this is having already made the Y98F mutation in it. So we would expect it shouldn't be binding everything. This had also been seen with other older antigens like the H2 that was circulating in 1957 to 1968 before the H3 pandemic came along. And we saw something similar, but not to the same extent with this Vietnam 2004 earlier bird flu outbreak HA. So what we saw is that the binding, this high background was actually not due to sialic acid binding. You know, if we treat the B cells with neuraminidase and desialate them, it doesn't decrease the background. The same thing was true with the H2 antigen also, where we saw this really, you know, untenable amount of background binding. But we got some clues from work that had just very recently come out in studying the H2 antigen, and also from some previous work that had come out a couple of years before, looking at some unusual flus that infect bats. And the finding there was that in the bat cases, for example, the HA of some bat influenzas doesn't seem to bind sialic acid at all in one case, or doesn't have it as the only receptor it binds. The bat flus also bind class 2 MHC on bat B cells, and that seems to be their main receptor for actually entering and infecting cells. So this paper, around the time that we were starting to do this work, had just come out that said the H2 virus HA that infected humans uses both sialic acid and MHC class 2 for cell entry. So naturally, this seemed to us like, you know, well, we should definitely check for these new bird flu H5 antigen. Maybe that's something similar is happening. And so, you know, Taylor and Oliver designed an experiment. Well, let's try just using a blocking antibody against class 2 MHC, HLA-DR, or one that binds more broadly against HLA-DR to PTQ. And what we saw is that that basically reduced the background binding to what's more typical for the B cell receptor recognizing on HA. So it was actually a nice resolution to this dilemma. And on the one hand, it gives us the tools to actually sort the B cells now that are specific for the new H5. But there's also some other interesting aspects to this. So, you know, we can show we can titrate the blocking antibody. It blocks it quite well, and it's sort of titratable away. And when we try other specific antibodies like anti-HLA-DQ, it doesn't block. Anti-CD74 for the clip, but that doesn't block. The same thing was true for the H2, so we sort of verified what was in the literature already. And the thing that was also surprising and interesting was that there really wasn't nearly the same amount of background by the older bird flu in 2004. So it's actually something that seems to be particularly having been selected in this new H5 virus that's been spreading so widely. And dairy cattle herds and many other mammals across the US. So knowing that this thing seemed to bind HLA-DR, we then were wondering, well, HLA-DR is, of course, a quite polymorphic gene, right? People have different alleles. I wonder if there's any effect of the allelic variance that somebody actually has in this binding. And of course, other questions, immediately come up with this, like, for example, what effect does it have on virus tropism? Is the virus able to be carried into other sites in the body because of binding HLA-DR and maybe binding to antigen-presenting cells or other cells that might be different than the usual sialic acid binding targets? So we devised some experiments where we had a bunch of samples from individuals where we knew what their HLA-DR genotype was, and we could do this experiment just of seeing whether the HA bound to their cells or not. And so what we found is, depending on the donor, so each of these rows is a different donor sample. These are the DNA counts of the H5 binding to them. This is replicate experiments for the two, so we saw quite reproducible binding or not, or less binding depending on the donor. When we summarize the data, what we find is that some people, based on their HLA-DR genotype, bind a lot of this H5 antigen. Other people have very little background binding for it. So we think there's a big effect of the allelic variance you have in your HLA-DR for this different interaction of the HLA or of the HA of the virus with your cells. So that's something that, in some ways I was very intrigued by this result. But it also, was sort of worrisomely reminding of conspiracy theories that happened during COVID, for example, that, the virus was engineered to infect some people and not others and this kind of thing. So, there's a lot of interesting potential implications of this. And we're now carrying out experiments to study this in larger cohorts of individuals with, wider range of HLA-DR genotypes, and especially people that are homozygous for a particular, allelic variant, so we can get the cleanest possible data. But I think it has a lot of interesting implications and would be something that would be of interest to this group, especially, here's hoping we don't actually have a pandemic involving descendants of the current H5 bird flu, but it's certainly possible, it seems. So anyway, just to summarize this last part of the talk. So we saw this background of binding with the H5s. It really seems to be blockable by binding MHC class 2. This is something that's been reported for a couple other flu variations, and there's actually some data that's not widely known that also seems like certain H3s may also have this similar property. So it actually might be more of an underlying theme for human interactions with the HAs of flu for certain particular virus types, not for all of them. And we find that there's an association between the background binding and the HLA-DR genotype is something that we need to follow up further, and this will be quite interesting to pursue. We certainly see that these HAs still bind sialic acid also, so it's actually not all HLA-DR. In this area, one thing that many of the labs that have been studying this topic of the HLA-DR binding by HA of flu is it's been difficult to identify direct binding with purified proteins. So people can detect it easily on a cell surface. You can block it with the anti-HLA-DR antibodies. There's some other data, like, for example, watching clustering of the HLA-DR when the HA is added. But we think that there may still also be some other binding partner or some other aspect that's not fully captured in the HLA-DR plus the HA data alone. So anyway, I'll leave off there, but really want to acknowledge the main people who did most of the work talked about here. So Oliver, Taylor, Artem, and then the initial B-cell sorting experiments I talked about, Katrina and Brandon, were the main ones responsible for that. And we'd like to thank our other collaborators and, of course, the funders of this work. And I'm happy to answer any questions, but thanks for your attention.
Questions for Scott?
Thank you for the exciting work. Now that you developed a multi-viral panels, I'm very interested to see if you'll get off-target effects from a vaccine, say for flu, stimulating cells that have been pre-exposed to COVID.
Yeah, so that's a good idea. Like, are there bystander activation of other specificities?
So it comes back to whether you vaccinate both at the same time or separately, et cetera.
Yes. Yeah, that's something. I mean, when we've looked at these flu vaccination cohorts, in more recent experiments, we've also had the other antigens present. So, but I don't think that we see strong evidence of a lot of bystander activation, but I can't say we ruled out that there's some potentially. Yeah, for sure.
Hi, Scott. No, that was great. Where do you think the Nipah virus specificity came from in that one spleen? It seemed
Yeah.
like from there was talk this morning
Right.
that that's really rare and it's only a certain part of Africa. I mean, that was just really interesting that you found.
So I think what the memory. So this is something that's come up in a bunch of projects that we've been involved in, is that when you look at the binding of antigens to memory B cells, I think that we do pick up sometimes lower affinity cross-reactive binding than you get if you make the purified monoclonal antibody and look in a standard kind of serology assay. Like, for example, there's some really clear data that are very nice about that for if you infect ferrets with an H3 virus, and then you later come back and infect them again with an H1 virus, you boost the H3 specific titers. And yet if you try to pre-clear or remove those H3 binding antibodies with the H1 antigen to begin with, at the level of the individual monoclonal, they don't seem to bind well enough to be removed that way, but they definitely get boosted. So, and I think that does happen in memory populations in general when people get vaccinated. The ones that actually are the B cells that are stimulated to become good antibody secreting cells, I think are only a fraction of all the memory cells that do get stimulated and may clonally expand, may get a bit mutated, but don't actually turn into real antibody producing factories. So yeah, but it's we think avidity is the reason for that difference because of the cell surface having so many BCRs.
Hey, Scott, great talk.
Hey.
3 things. Your panel's already pretty big, but we have a lot of really well-characterized antigens. So if there's something else you need, let me know.
Thanks. Yeah, no, we'd love to try. Yeah.
Number 2, your data set, it's already absolutely incredible, but it'd be super cool to actually get epitope specificity during screening.
Yes.
So like one way to do that would be to knock out certain epitopes by redesigning the surface.
Yep.
We're doing a lot of this with HA in particular. So
yeah, that's superb. I mean, that's a direction that we've started going down as well, but I think it'd be great to talk and put our heads together because yeah, that would be even nicer if we, in the initial data set, could say, okay, these are the site A binders on the hemagglutinin head. These are the stock binders.
And maybe like split the sample or something and make so that you just have a bunch of variants of H1 where you map the epitope specificity and then you have your broad panel, which would be cool. And then number 3, the data as they are, but particularly if you can get the epitope specificity, would be very valuable for training machine learning models for antibody antigen prediction.
For sure.
So if you're able to share the data, that would be wonderful.
Yeah, no, those are things we're keenly, interested in as well and beginning to model. But we'd love to collaborate and discuss further. Yeah.
You've thought about this a lot, obviously. So it seems like that receptor would be quite restricting in terms of cell tropism, and cells that express class 2 really don't, they don't bud infectious virus, right? So what do you think about that?
Yeah, so we're not sure yet whether it's necessary for the virus to infect. We know there's evidence from the H2 and the bat data that it can definitely be permissive to allow the virus to infect a cell or enter a cell. I think it could well be related to tropism of the virus, where it can be carried, for example, into the central nervous system, which is true for some of the flu types and not others. And it's definitely a feature of the H5, the recent bird flu, when it infects things like cats or other animals. So I think I think these data don't suggest that it can't infect an epithelial cell, but it might just be able to infect a wider range of cells, and some of them that may be more mobile and may result in more spread or something. No, the current H5 does bind salic acid as well. So it's more similar to the H2 in that regard. But the bat ones don't seem to need salic acid, and so that is a difference. Yeah.
Any other questions? All right.
Okay,
Thanks,
thank you.
Scott, again.